Hello students, today's class will be Development of Dentition, Odontogenesis, Part 2. Part 1 video of Odontogenesis, please kindly see that and come to this part. Moving on. In this class of Odontogenesis, we will be covering the following topics. Please make a note. Hertwig's epithelial root sheath and root formation, epithelial mesenchymal interactions, histophysiology, molecular insights in tooth morphology, and MCQs. The development of root begins after enamel and dentin formation has reached the future cemento-enamel junction. Now, if you can see in this diagram, the blue colored area is the enamel, the pink colored area is the dentin, the tall columnar cells are inner and enamel epithelial cells are given, and when the enamel and dentin reaches the future CEJ, root formation proliferates downwards. Epithelial diaphragm. You have to know something called as the epithelial diaphragm before we proceed to root formation. Prior to the beginning of root formation, the outer enamel epithelial cells and the inner enamel epithelial cells of Hertwig's epithelial root sheath bend at the future cemento-enamel junction into a horizontal plane to form the epithelial diaphragm. If you can see in the previous diagram, when enamel and dentin reach the future CEJ, root formation starts. And when root formation starts, what the inner enamel epithelial cells do is, they bend at horizontal angle, they bend in a horizontal plane at almost right angle to the root forming structure to form something called as the epithelial diaphragm. Differential growth of epithelial diaphragm causes the root trunk to divide into 2 to 3 roots. When a tooth has 2 or 3 roots, that is because of differential growth in the epithelial diaphragm. This is a one word to note. Differential growth of epithelial diaphragm causes the root trunk to divide into 2 to 3 roots. Coming to Hertwig's epithelial root sheath, or HERS, we'll refer to it as HERS. The enamel organ proliferates down to form the Hertwig's epithelial root sheath. Function of the Hertwig's epithelial root sheath is it molds the shape of the roots. It has only 2 types of cells, inner enamel epithelial cells and outer enamel epithelial cells. What happens is, in the Hertwig's epithelial root sheath, inner enamel epithelial cells induce differentiation of radicular dental papilla to form odontoblasts. Odontoblasts, what they do? They lay down dentin. After dentin is formed, following this, the inner enamel epithelial cells are differentiated into cementoblasts and they lay down cementum. Just like how in the crown, we saw first dentin is laid down followed by enamel. Here in the root, dentin is laid down first followed by cementum. In the same image again, you can appreciate the Hertwig's epithelial root sheath proliferating vertically downwards to form the epithelial diaphragm, after which it further proliferates downwards. After the first layer of dentin is laid, Hertwig's epithelial root sheath loses its structural continuity with the root surface. If the continuity of HERS, Hertwig's epithelial root sheath, is broken prior to dentin formation, it leads to formation of accessory canals. If the before dentin formation, in this flowchart, if you can see, Hertwig's epithelial root sheath stimulates inner enamel epithelial cells to form odontoblasts to lay down dentin. After dentin is laid down, connection of HERS with the root surface is lost. But if the connection between the root surface and Hertwig's epithelial root sheath is lost before the dentin is formed, it will result in accessory canal formation. After disintegration, remnants of the Hertwig's epithelial root sheath passes near the external surface of the root. These remnants are called as cell rests of molasses. Now, in the first part of odontogenesis, we saw another remnant, that is the cell rests of serrae. Remnants of the dental lamina are called as cell rest of serrae. Remnants of the Hertwig's epithelial root sheath are called cell rests of molasses. Moving on to physiological stages of tooth development. Morphologically, stages of tooth development are divided into bud stage, cap stage, and bell stage, depending upon the shape of the enamel organ. Physiologically, stages of tooth development are initiation, proliferation, histodifferentiation, morphodifferentiation, and opposition. There are many important MCQs based on these concepts. So, highlighting on those MCQs will move the class further. If you can see, morphologic and physiologic stages are given. We have to correlate which morphologic stage will be more or less equal to the physiologic stage. When the morphologic stage is only in the dental lamina area, or it shows only the dental lamina, physiologically it is correlated with the initiation stage. Bud stage and cap stage of morphogenesis correlates with the proliferative stage of physiologic development. Early bell stage correlates with the histodifferentiation stage, and advanced bell stage correlates with the morphodifferentiation stage. When the hard tissues, enamel and dentin are laid down, it correlates with the opposition stage of physiologic development. I'll come again. Morphologic stages and physiologic stages are given. MCQs will be based on which physiologic stage correlates with the following morphologic stage. Dental lamina correlates with initiation, bud stage and cap stage, proliferation, early bell stage, histodifferentiation, advanced bell stage, morphodifferentiation, formation of enamel and dentin with opposition. Now, in these physiologic stages of tooth development, if you have any kind of disturbances, it will result in tooth anomalies. Any result of disturbance in the initiation will result in anodontia, supernumerary tooth, or a dermoid cyst. If there is any disturbance in the proliferative stage, it does not affect the tooth. In histodifferentiation, if there is any disturbance to the tooth germ or the tooth structure, it can manifest as amelogenesis imperfecta, dentinogenesis imperfecta, dentin dysplasia, regional odontodysplasia, atypical dentin or osteodentin. This atypical or osteodentin might result due to vitamin A deficiency. Morphodifferentiation stage can result in the following 9 disturbances: talons cusp, twinning, loss of a cusp or root, mulberry molars, Hutchinson's incisors, densin dentin, macrodontia, fusion, and gemination. Apposition will result in any disturbance in apposition stage might result in enamel hypoplasia, enamel hypocalcification, intrinsic straining, and concrescence. Something else to notice in morphodifferentiation, not only macrodontia, if morphodifferentiation stage is disturbed, macro or microdontia can result. Molecular insights in tooth morphogenesis. Pay attention because this is a completely MCQ based part. You will be given the following genes and asked to which factor of morphogenesis in tooth development it correlates. Please note down, mediators in enamel knot formation is mediators in enamel knot formation is by FGF8 and SLIT1. Genes important for tooth formation to take place, FGF9 and PAX9. Inhibition of tooth formation in interdental areas, gene responsible, BMP4. Earliest mesenchymal marker for tooth formation is LHX6 and 7. Earliest epithelial marker for tooth formation is FGF8. Previously, we saw earliest mesenchymal marker for tooth formation. This is earliest epithelial marker for tooth formation. Marker for tooth agenesis, that is absence of tooth to develop, MHX1 and PAX9. Epithelial proliferation or tooth initiation is mediated by sonic hedgehog, SHH gene. It is also seen in OKC, odontogenic keratocyst. Morphogenesis and histodifferentiation of enamel organ is mediated by CBFA1 gene and RUNX2 gene. Location of the developing tooth germ, genes responsible FGF8 and PAX9. Tooth patterning is done by PITX2, gene PITX2. Bud stage to cap stage is controlled by the gene PAX9 and activin beta A. Thickening of the epithelium is controlled by gene BMP7. That's all for this class. MCQs. Atypical dentin results due to disturbance in which of the following stages of tooth development? Like I'd already said, atypical dentin or osteodentin occurs due to vitamin A deficiency. It occurs in which stage of tooth development is the question. Options: histodifferentiation, morphodifferentiation, proliferation. Answer is option A, histodifferentiation. Second one, earliest epithelial marker for tooth formation is dash. Earliest epithelial marker is FGF8. Students, always remember, molecular insights in tooth morphogenesis is a one word. Here in this slide, I have listed all possible molecular insights. So kindly make a note of them and by heart them. Thank you.